Welcome back. In this section, we are going to go through the 4 steps of mitosis. So first, what is mitosis? Well, mitosis, what we have done is we have taken a cell and we have done one chromosome duplication. Okay, that already happened in interphase, one chromosome duplication. And in mitosis, we are going to do one division. So we've gone from one chromatid to forming a chromosome that has sister chromatids. And now they are going to be divided to 2 daughter cells. Each daughter cell will be identical to each other, and each daughter cell will be identical to the parent cell before that chromosome was duplicated. So there are 4 steps to mitosis, and they always occur in the same order. So you're going to need to know the steps as well as what happens in each step. Recognize pictures of each step. The first step is called prophase, as in before the stuff that is happening. Metaphase, stuff is happening in the middle. Anaphase, we're away from the middle. And telophase, we are ending. Think of telophase kind of as the opposite of prophase. So let's get to it, shall we? Of all the steps, the longest phase is prophase because a lot of stuff has to happen. The first thing that happens is that those 5-foot-long strands of chromatin need to condense. They need to go around the stones and twist and condense and become those visible chromosomes. In addition, that nucleolus paper, the nuclear envelope starts breaking down. It's going to completely disappear. And the centrosomes, because remember they were duplicated as well, they are going to move to opposite poles of the cell. Okay. So whether you want to call it north and south, east and west, it doesn't matter. They're just going to be opposite from each other. So coming from the centrosomes, we are going to start seeing some cytoskeleton. These fibers that we're going to call spindle fibers are going to be leaving the microtubules of the centrioles that are located in the centrosomes. Okay. So if we look at a cartoon drawing of what is happening in prophase, we can see a lot is going on. We see 2 different centrosomes and we see spindle fibers coming from them. We see a nuclear envelope that is polka dotted showing that it is disappearing, and we see X-shaped chromosomes showing that they have been duplicated, attached in the centromere in the middle. However, if we look at a photomicrograph, remember, we can't see a lot of that detail with a light microscope. So what we are going to see is the absence of a nuclear membrane, and we are going to see visible chromosomes. And they look like a bowl of spaghetti because they are not organized. And that's what prophase looks like. All right, so after this, we are going to move to metaphase. In metaphase, what's going to happen is the chromosomes are going to move to the middle of the cell. And so things can really move around now because there is no nuclear envelope confining things to a certain location. So remember, we've got 46 X-shaped chromosomes with sister chromatids. And so they are going to line up single file right down the middle. And notice the way they line up is so that wherever the centrosomes are on the side, the X is so that one sister chromatid is facing one centrosome and the other sister chromatid is facing the other one. Okay, so you can see that on both of these drawings. And the reason for that is those spindle fibers, which are coming off of the centrioles, are going to be attached to the centromeres of each of those sister chromatids. So they attach like a belt at the middle of it. Okay? And so the centrosome on each end attaches to the sister chromatid on that side of the X chromosome. So you can see what's going to happen here is that X chromosome is going to be tugged at two opposite sides, but not in metaphase. In metaphase, they line up in the middle and the spindle fibers attach to the centromere for opposite poles. So what does this look like in a photomicrograph? Well, you've got centromeres lined up and you actually can see spindle fibers in them. And even though we show this beautifully in our cartoon drawings, when we look at the chromosomes in a photomicrograph, they don't look nearly as neatly lined up as our drawings do. All right, so the first phase was prophase, the second phase was metaphase. Now we're ready for anaphase. And in anaphase, those spindle fibers are going to start tugging. And so they're going to start tugging towards the opposite poles. So the chromatids are going to be pulled towards the opposite poles by the spindle fibers, and they're being pulled by the centromere. So the centromere is leading toward the pole. And as you can see, the top and the bottom part of the chromosome is trailing behind it because that's not where the pole is. Okay. At the same time, in anaphase, the cell starts elongating, more space between these two sets of sister chromatids. So the sister chromatids have separated, okay? They don't line up like this drawing shows here on the right. The blue cell is factually inaccurate because the pull is from the centromere. So they're going to look more like the other three cells shown up there. All right, so here's photomicrographs to show you. The purple cell is showing you at the beginning of anaphase and then later in anaphase. The middle is just showing one where you clearly can see kind of like the U shape as the sister chromatids, which were like this, are getting pulled away from each other. And then there's two more cells on the right, one in pink and one in black and white, showing you that the cells are starting to get elongated in shape because we need to start giving more space between the two sets of what used to be sister chromatids. And then this is going to bring us to our fourth phase of mitosis, which is telophase. And the first thing that's going to happen in telophase is the mitotic spindle is going to break down so that it's no longer going to be attached to the chromosomes. And the chromosomes now are at opposite poles and they're allowed to decondense back out to form chromosome 10. So they're no longer going to be visible chromosomes. They're just going to be chromatin. At the same time, the nuclear envelope is going to start reforming. So basically, this is going to be the opposite of interphase, and the cell is preparing to divide. And sometimes you can see that a little bit of narrowing, which is a different process, which we're going to talk about shortly. So here are three photomicrographs. You can still see just dark blobs representing where the chromosomes are. You can see a little bit of spindles left behind, but you can see that these cells, this one big cell is starting to get ready to divide. It really looks like it's trying to become two cells, okay? So I'd like you to stop here, look at these pictures, name each cell and each step in each cell, tell what happens to the DNA. Do we have chromatin? Do we have chromosomes? What's happening to the nuclear membrane? What's happening to the centrosomes? What's happening to the centrioles? Okay. And with that, thank you so much for understanding the mitosis and then by talking yourself through it without the pictures as a prompt. And I will see you soon so we can talk about what is happening at the end of mitosis.